My name is John Constantino. I appreciate the opportunity to participate in this course for UCLA. And the title of my talk is Genetic Epidemiology and Pathways to Equity in Autism. This is my disclosure of interests. I have a small advisory role with Cognoa, which is a electronic phenotyping company. No stock equities. I receive royalties on the social responsiveness scale, which many of the people in this audience will recognize that. And I receive research support from these donors that are listed on the slide. Well, what I'm going to do today is take a quick whistle-stop tour through genetic epidemiologic research and related research that has direct bearing on health disparities. And the first, which again will be familiar to the UCLA audience because of the close collaboration with Jenny Lowe, Dan Geschwin, and many members of the gene discovery team in autism there, which is a paper that issued from a gene discovery project in African-American families affected by autism that is listed on this slide. And it is about a very serious health disparity on the basis of race. And that disparity is actually depicted on this slide. These are sort of graphics from the two most recent surveillance years of the Centers for Disease Control's Autism and Developmental Disabilities Monitoring Program, ADAM. This is 2018 and this is 2020. And the significance of those two surveillance years is that in the community at this point, the diagnostic prevalence of autism as established by clinicians and schools in the community is approximately equivalent for all races in the population. This was not always the case. Historically, it was the case that minority children were less likely to be at least recognized or diagnosed or officially sort of receiving a community diagnosis of autism. And for a very long time, it had been assumed that that was the reason why minority children who were affected by autism appeared to have more severe syndromes. It was the idea that, well, if we're only picking up the most severe cases, that is the reason why minority children are on average more severely impaired. But the remarkable story from these two surveillance years of the Centers for Disease Control is that in these years in which finally the prevalence has equalized as a function of race, there remains an extraordinary disparity, here illustrated by the difference between white girls and boys and black girls and boys in the proportion of children, if they have autism, that are also affected by either borderline intellectual functioning or intellectual disability in the range of IQ less than 70. And here is the key statistic for that, that for girls, here you see a rate for African-American girls of over 50% for intellectual disability compared to the mid-20s for girls who are white, and for black boys, 45% or close to it in comparison to Caucasian boys, just over 20%. And this is related to a condition, of course, that affects 1% of the children in the population. And this remarkable disparity is something that we have no idea what the cause of that is, or had no idea what the cause of it was when this was published in 2018. People had lots of perspectives about that, but the nature of this particular analysis was to go within the African American population and look at the predictors of this particular outcome to see if we could get some idea of what the cause of this was and what we can do about this extraordinary and lifelong and severe disparity. Well, what we did was to look at the pathways to diagnosis among 584 children in the gene discovery program for African American families affected by autism. And we utilized data from this long-standing study. It's now in its 13th year, led by Dan Geschwin at UCLA. And this particular implementation of an interview was about ascertaining how children with autism among African American families were diagnosed, when the parents expressed concerns about the children's development, the various demographic and socioeconomic factors affecting the family, the timing of diagnosis, the services that were started, the interactions with service systems, and the overall odyssey to getting a diagnosis. And what was learned from that from the analysis of that data was very revealing, in that the mean age at which parents first reported concerns about their babies who ended up developing autism was around 29 months, but the mean age of the diagnosis itself didn't come until 42 months later on average, at close to 65 months of age. And again, this was even after the parents had shared concerns in most cases with professionals about their children's developmental progress. 97% of the families recorded having a doctor or healthcare provider. 98.5% reported having health insurance with a mix of public and private insurance. And one of the sort of very sad kind of markers of the odyssey of the families was that fully 15% of the children affected by autism had to have visits with more than 6 professionals before that diagnosis was made or confirmed. And of course, this was part of the delay in going from the parents' expressions of concerns to a diagnosis. One of the other things that we were able to study in this sample, because of the genetic nature of the study and the family studies kind of design, was that we were also able to ascertain the outcomes of the children, particularly their cognitive outcomes, as a function of the cognitive outcomes of their family members. So looking at the mothers and fathers' IQ, the IQ of siblings, and then a number of demographic factors related to the diagnosis. So the age in which the children were diagnosed or received first services. And this was an indication that it was true that some of the earlier identified children were more severe and more severely impaired, but there were really no associations in this particular analysis with the other usual suspects for the causes of cognitive disability in minority children in general, or the disproportionate burden of intellectual disability in children who are in, for example, stressed socioeconomic circumstances. So neither education level of the parents, family income, the and the characteristics, as I said, of the family members with respect to IQ, and nor did the level of severity of the autism itself really have any significant bearing on the cognitive outcomes of the kids who were enrolled in this study. So this is a correlation table that looks at those associations, and these analyses were also done through hierarchical regression methods to show that, again, that the things that one would expect might predict cognitive impairments of the children being disproportionately affected by intellectual disability burden among the African American population when they have autism were really not predictive in this sense. And so the conclusion from this particular analysis was that these particular kinds of excuses for disproportionate cognitive burden among these children really did not apply, and that the overarching delay in diagnosis and the very severe kind of disparity in access to services faced by African American children appear to be very significant contenders for which we kind of bear the burden of responsibility and proof that resolving those would not level the playing field for these particular this particular population of children. So, so one of the reasons why this is so important is that there are at least open trial types of indications that the intensity and duration of developmental therapy services that are specific to autism, when applied early enough in a child's development, actually does have a payoff in terms of these dimensions of functioning in terms of adaptive outcomes. So social adaptation, play, motor skills, language and executive function, and overall adaptive and academic skills are seeming to be improved to some degree by the developmental therapies for which, as a population of children, these kids have almost no access across almost the entire sample on the basis of looking at the long delays and the late timing of diagnosis and the again the extent to which these children manifested a general sort of disproportionate burden of intellectual disability superimposed on the condition of autism. And so these data and the analysis that chipped away at the sometimes suspected causes of these cognitive disabilities that could not explain that variation in the African American population of children with autism raised the specter of these delays, not only in diagnosis, but in the access to services as a form of structural racism. And that this particular kind of data set documented the ways in which racism and racial inequity are playing into these very significant disproportion of intellectual disability outcome. And so an editorial was written in response to our paper independently, in which that was emphasized that a third of the parents cited availability of professionals as a barrier to diagnosis, and that tracking back into the problems with an available workforce that's limiting capacity to access to the kinds of professionals who can make diagnosis in a timely manner and deliver therapeutic services in a timely manner is a real problem, as well as the sort of knowledge that recent workforce surveys have shown that only 2% of developmental or behavioral neurodevelopmental pediatricians are African American in relation to this particular racial disparity. And the authors felt that these data that were compiled from that data set, and along with previous work in inequity and autism spectrum disorder, I'll show that in a second, necessitate an immediate evaluation of how these issues of structural racism have impacted ASD diagnosis, and an action plan was called upon, to address this inequity, that we really bear the burden of proof that resolving these particular kinds of inequities would not be capable of resolving that very severe disproportionality in cognitive disability outcomes. And they ended their editorial with the question of whether we as an autism clinical and research community have the will to achieve this goal. In following this up, in this 13th year of this ongoing project, and having known that this was the way the data were laying out a number of years ago, we had decided in the most recent renewal of this program to concentrate on earliest possible diagnosis for these children, hoping that if we could use the research study in which we were enrolling African American families affected by autism. We could use the research study as a lever to speed up diagnosis among African American children in the community, that we could then also observe whether or not they would have better outcomes, potentially by virtue of having early, timely access to intervention. And thus far, we've enrolled 70 African American toddlers in this more focused data collection. 85% of them have ASD. For most of them, the research study provided them their first diagnosis. So these were children who were referred by their parents or by their primary care doctors, and had never, for the most part, had never been diagnosed with autism and received a diagnosis of autism. And in this particular project, at a mean age of 27.9 months, which is over 3 years sooner than the national average. And those children were not diagnosed 3 years later or sooner than the national average because they were more severe on average than our, all comers, over 500 kids that we had enrolled at various ages into the prior gene discovery project. So if you look at the mean scores for their composites for adaptive functioning and communication and socialization, what we found was that the standardized scores were really no different for these toddlers, the ones that we've been able to fully assess so far, than the whole population of African Americans that we had been enrolling over 13 years of time. So this study was picking up the kids who were equally impaired to the whole population of kids, African American kids affected by autism, but speeding up their diagnosis by 3 years. And the problem was that we found very quickly was that just endowing them with an earlier diagnosis didn't get them any more services than our group that had an average age of diagnosis of 65 months got. So typically our children in this particular program, we were very dismayed to find that even after we handed them the diagnosis using all of the standard diagnostic instruments, that they were still getting less than 1 to 2 hours a week of therapy for their condition. And many of these kids were recruited in what have been previously described as service deserts, where even just geographically defining where the children live puts them in a situation where they're at risk not to have much access to the appropriate kinds of services. And so they at intake, for behavioral therapies, for example, specific to autism, they were getting a mean of 1.4 hours a week of therapy even after we diagnosed them. We were able to boost this on the basis of a grant which allowed us to provide half of the children with a sort of more standard, even if somewhat minimal, complement of autism-specific therapies, to get the group average to about close to 5 hours per week. So it was basically a group of kids that got closer to 8 or 9 hours a week, that of half of the kids that got that comprised that particular average. And then, of course, the COVID pandemic hit, and that wiped us out and put us back to where we started. And unfortunately, and we watched kids who had made some gains with those behavioral therapies go backwards in that context. And more on that as we resume intervention and try to recapture some of those gains. But this is a little girl who we first saw before the COVID pandemic, who was at the first percentile for all of these domains in the Mullen scales of early learning. And we honestly thought that she was very likely to not only have ASD, but global cognitive delay. She was entered into this program and stalled out during COVID, and her progress stalled. But post-COVID, she was re-enrolled in the intervention. And at this stage, now she's 4.5 years old. She is in the range between 30 and 50th percentile for all of these skills. So she's sort of a success story, even in a child who we would predict might not respond so well to developmental therapies because of the suspicion of cognitive impairment from the very beginning. And as has been pointed out, that there are ways in which just knowledge and recognition by families of the likelihood of the development of autism and the judicious implementation of therapies. There are other lines of evidence that show that these children's outcomes and adaptive functioning can be raised by close watch and early application of intervention. And I think these children are no exception. And like I said, in the African American community, it is as if, substantive intervention really does not get going until school age. And unfortunately, that situation is associated with this wholesale shift in the cognitive outcomes of that population of children. So I want to move on to just a couple of other areas of equity that people don't necessarily think about as issues of health equity, but that are important in autism and that are addressed in various ways by genetic epidemiologic research. And the first one that I want to point out is that historically, in recent history of genetic research on autism, there's a great deal of interest in autistic syndromes that can be traced to a highly parsimonious biological cause, the monogenic syndromes. And there's a lot of excitement and there's a lot of appropriate allocation of scientific resources towards children and families affected by monogenic syndromes causing autism. And the reason for that, of course, is that monogenic syndromes that cause autism have this sort of biological tractable structure that can be pursued in the laboratory and that we would hope would engender insights for all children affected by autism. But one could ask also whether the pendulum for the allocation of research resources for autism may have swung a little too far in the direction of monogenic syndromes. And there are some things about monogenic syndromes that are not so representative of the whole population. So first of all, the monogenic syndromes that cause autism are typically on the basis of single gene defects that arise in the germline. It's conditions that children have, but their parents don't have. They're discovered by comparing the genomes of the affected children and their unaffected parents. And they are sort of single hit smoking guns that cause autism, but they, by that definition, the fact that they are germline conditions don't account for the heritability of autism, because autism, and when you look at most cases in the population, most of the autism in the population is inherited, and most of the monogenic syndromes are not inherited, and so therefore not representative of all the cases. There are other things about monogenic syndromes that are not representative of all cases also, which is that typically monogenic syndromes that result in autism also almost always are associated with intellectual disability, whereas the majority of the cases of autism that are inherited in families, which is most cases of autism in the population, are not accompanied by intellectual disability. And this has led people to conclude that that even though we think of autism-specific genes because of the association of these monogenic conditions with autistic syndromes, it's not so clear that those are more about autism than they are about intellectual disability with autism. And so the hope, of course, is that the monogenic syndromes are going to lead us to biological insights into all, cases of autism, and particularly the common inherited ones. And there are reasons to believe, because of overlaps in many branches of medicine, that the Mendelian disorders that are caused by these kind of rare, highly deleterious mutations are related, to the causes and biological mechanisms for complex diseases. But the point here is to say that that there needs to be focus and equity in the study of inherited forms of autism. And historically, there has been some degree of concern about investment in that, because polygenic risk and inherited risk is complex. It's more difficult to get a biologically, kind of tractable, kind of get your hands on that. But what I want to point out is that recently, because of genetic epidemiologic research, we actually have identified some potential convergent pathways for polygenic risk that may make it tractable, not on the basis of a specific genotype, but on the basis of specific developmental phenotypes that would allow for discovery within this particular subset, large subset of all autistic syndromes, to lead us to important insights for treatments for children. So what do I mean by that? And many of you have heard me perhaps talk about this before, but inherited autism, on the basis of data that we have amassed over the years in looking at how autism runs in families and what gets transmitted in families and what aggregates in the families of individuals with autism and what is and isn't inherited, about the early appearing predictors of autism and autism recurrence in families, is that it appears that there are sets of polygenic risk that converge on specific highly heritable developmental phenotypes. And I won't go into the details about that in this talk, because this is a talk about disparities, health disparities in relation to autism. But what I'm trying to do here is to make a case that that polygenically inherited forms of autism are biologically tractable in a different kind of way, and that we are now able to are starting to be able to type young children and their risk for autism or their risk for autism recurrence within families affected by autism on the basis of a pretty short list of developmental endophenotypes that account for a great deal of the variance in whether autism is going to recur in that baby or not. And what I have illustrated here is the highly heritable developmental endophenotype, let's call it, of social visual engagement or disengagement that can be identified and ascertained by eye tracking methods as early as the 6th month of life. We can now identify aspects of autism-specific background, inherited liability that's indexed by the quantitative phenotypes of parents that can be measured, before a baby's born or the day that a baby's born. And we're starting to be able to identify motor coordination and ADHD-related phenotypes that we believe actually contribute to the matrix of polygenic risk for autism, and that autism represents an amalgam of pathways that can arise from different permutations or combinations of different extremes of liability across these different traits, each of which is highly inherited, each of which is independent from the other, each of which is fully continuously distributed in the population. And therefore, that when we do studies of comparing people with autism versus controls, one of the things that this infers is that if we're not measuring these same traits in the controls, and assuming that the controls are just blank slates of non-autism, we're missing a lot of opportunity to understand differences between people with autism and controls. missing a lot of statistical opportunity to identify biomarkers if we are not recognizing that these particular contributors to inherited forms of autism are floating around all over the place in the general population. And we have to get serious about measuring them when we're looking for things that differentiate people affected by autism versus controls. And so this particular kind of way of thinking about autism, coupled with the notion that as one becomes more and more affected by a clinical level of impairment of autism-related traits, interestingly, the severity of the condition becomes less and less affected by those genetic contributors and more by what we believe are just sort of stochastic influences that affect severity. And so we're missing opportunities to identify, to study polygenic autism in a biologically rigorous way. Number one, if we're assuming that the controls are unaffected. Number two, if we are thinking that severity of autism after one crosses the clinical threshold is determined by the same factors that cause autism. And we have this paper, which is a compilation of prior empirical studies, makes the case that those two assumptions about how we study polygenic risk and how we look for biological markers may be wrong. And so we may be selling polygenic autism short and need some new ways to think about it and study it to make it as tractable scientifically as monogenic syndromes. Now, what do I mean by that? Well, this is a diagram that was produced in 1972 by Arthur Guyton to lay out this kind of idea for hypertension as a different kind of quantitative clinical syndrome. And what it does is it depicts Guyton's understanding at the time of the various levers of blood pressure. So one can think of blood pressure as a singular quantitative trait that's easy to measure. If it gets too high, it's hypertension. But really, it's an amalgam of a dynamic set of checks and balances, capillary membrane dynamics, muscle blood flow, stroke volume of the heart, different aspects of electrolyte balance throughout the system, heart rate, pulmonary dynamics, that not only are these different components of the condition, of the contribution to blood pressure, but they actually compensate for each other. They actually interact with each other in different ways. And the question is, what if the genetics of autism is really like this? That there are these different kinds of developmental contributors to the condition, each of which has its own role, each of which is in dynamic balance with the other. And if we're trying to understand the genetics of the whole condition without breaking it down into these component parts, we'll get very confused, because there are some people who have hypertension as a result of issues pertaining to stroke volume, but others that have to do with electrolyte imbalance, and others that have to do with capillary and vascular dynamics. And they're all in that same pool. And so thinking about autism as a condition which is variously contributed by these different combinations of liabilities is a different way of thinking about how to get to the biology of it, and in fact, how to intervene. Because to intervene in blood pressure, one kind of intervention doesn't fit all. If it's a stroke volume problem, you have to use a medicine or a treatment that's going to address that. If it's an electrolyte problem, you have to address that. If it's a capillary or vascular dynamics problem, you have to address that. Those are three different things, and they're treated in three different ways. And so to kind of put this whole kind of idea together, just a comparison that we, this is a figure from another recent review of this, but we've thought about autism as this kind of fractionable triad of symptomatology of the different symptoms that comprise the condition. But what we're starting to think is really happening is that it's contributed by things that don't look like autism at all. And that autism, when it develops, then results in these highly intercorrelated sets of traits that are really a convergent kind of syndrome that arises when typical development doesn't go well. And that the symptoms of autism end up being a kind of epiphenomenon after a developmental derailment has occurred on the basis of liabilities related to a number of different fundamental human traits, extremes of which, again, can contribute to the development of the syndrome, or at least we've already shown now aggregate in the families affected and the family members of affected individuals and predict recurrence in these sort of very intriguing ways. So more on that another time, but the point there is we should think about new approaches to making the more common forms of autism more tractable to science and also potentially more tractable to interventions that are focused on moving these liabilities into the normative range so that novel opportunities for pre-diagnostic intervention might be highly informed by understanding these developmental pathways within an individual patient, knowing that an individual patient may represent his or her own unique amalgam of these extreme traits that would contribute liability to the condition. Okay, a couple more to talk about. We know that sex is inequitable in autism. This has engendered a theory about the possibility that females may be relatively protected from autism in relation to males. That has been very difficult to prove. The reason that theory has arisen is because most of the genetic liability for autism, whether in the realm of polygenic risk or in the realm of rare variant risk from de novo mutations, is mostly on autosomes, not on sex chromosomes. And so it's believed then that therefore, given that the sex difference of 3 or 4 to 1 exists in all cases of autism, can't be explained by X or sex linkage, that there must be something about female sex or sex-specific sensitivity to genetic liability that lies across the autosomes. And the female protective effect has been very difficult to demonstrate because it's been difficult to demonstrate the existence of the Carter effect. That is to say that females affected by autism would be expected, if they are relatively protected, to have a higher burden than the average male, and therefore to have a higher likelihood of a recurrence of autism within their family members. And there have been some studies that have suggested this, and some new ones. There was one just posted in bioRxiv to this effect. But we decided to kind of test this in an important way that has to do with the another implication of the female protective effect, if it exists, would be that the sisters of individuals with autism, the unaffected sisters, might be very concerned that if they are harboring an enhancement of genetic risk, that they would have an inordinately high likelihood of transmitting autism to their sons. And so we were able to essentially take data from the Swedish National Register, where there was 2-generation genetic epidemiologic ascertainment of autism across the generations with confirmation of cases in the offspring generation, and to ask the question of what is the actual estimate of risk to sisters offspring. And it turns out that in this marvelous epidemiologic sample, that the sisters, for sisters of ASD affected individuals, the risk of autism is only about 2 to 3 times the population risk and not significantly elevated over that of unaffected brothers. of an autism proband. And so it's not so much that it seems that females are protected over the population average, so much as that males are more sensitive. And although that may sound like a mincing of words, it has very important implications for the absolute risk to the unaffected females, which in this study indicates that it's about what you would expect from the general second degree relative risk for autism, and not inordinately elevated among those sisters, and in some ways a general kind of relief that that it looks like the risk to their offspring is not as high as would be predicted by a specifically female protective effect. And this was an editorial written about that, and how important genetic epidemiology is for answering questions that are otherwise very difficult to answer. And I really recommend it to any students of genetic epidemiology, because it talks about, it likens genetic epidemiology as a field to Galileo's use of the telescope. It's a lovely editorial, and quite an affirmation of some of the leaders of the field of genetic epidemiology that I think, as a student of that field for my whole career, I've been very grateful for some of those leaders. Now, a last couple of comments, and then I'll wrap up. There is another kind of equity to deal with, even though I said that, you know, kind of polygenic autism got the short shrift over rare variant autism in general in a lot of our allocation of scientific resources. But if you're part of any single type of autism, or any ultra rare, you know, familial condition for autism, you know, it's one thing to talk about all rare conditions and kind of how, you know, the scientific resources is entrained on monogenic syndromes, but if you have any one of those rare syndromes, you know, you have a disparity in the sense that even if it has been established as a cause of a condition like autism, it's so rare that it's very difficult to amass adequate resources for, you know, the kinds of treatment and understanding and clinical trials and so forth that all of us struggle with in terms of working on rare conditions when that's part of our portfolio. And to be clear, rare conditions are certainly a part of my own research portfolio. I don't mean to dismiss them or set them aside. I think we have to use all of the information and all the biological information from these conditions, and ardently hope that this will lead to you know, breakthroughs in understanding treatment. But for any one rare condition, it's a real problem, and what we have exponentially encountered in the realm of clinical genomics is there's all kinds of rare conditions for which we have no idea whether it's the cause of a condition or not. So this is a plot of essentially, I cut off the, well, you can see it in the legend for the table, of the percent of variations of uncertain significance that have been uploaded to ClinVar over the years. And as clinical genomic characterization of patients in general in medicine has, you know, rapidly accelerated because of, you know, reduction in cost and so forth, we're learning that when we clinically genotype a lot of our patients, we end up with rare variants identified that we don't really know if they're pathogenic or not. And that family is stuck because it hasn't been established in science whether that particular variant that they're carrying, that their child or their loved one is carrying, is associated with these conditions. And therefore, they may be, you know, under the most, you know, profound kind of disparity at all of being an N of one against, you know, versus an army of other kinds of, you know, pathways to science, and who's going to listen and look at their condition and incorporate that into the discovery process. Well, what we have decided to try to do this is a collaboration of all of the intellectual and developmental disabilities research centers, of which UCLA is one of the 14 or 15 of those active centers. And what we hope to do in this study is to take all of medicine as our playing field and try not to waste any information from any person who has been clinically genotyped as a function of medicine. And lots of times when any patient, whether they're in a diabetes clinic or a developmental disabilities clinic, gets a clinical genetic test, as I was pointing out, they'll have a variant of unknown significance. And what we wanted to do was couple these variants of unknown significance with information that may help us accumulate all of the data and put together genotype and phenotype in a way that we can accumulate a resource for gene curation and recognizing what that association looks like. And so the way that we're doing that is to essentially co-register electronic health record data of the patients, their actual genetic variant that whatever their variant of unknown significance is, all of their electronic health record data that might be relevant to helping to understand the association between genotype and phenotype, and when possible, also doing a virtual rapid phenotyping of these patients so that instead of what currently exists, which is a clinical genetic test result from a reference lab that is only associated with a diagnosis that was the presumed indication for genotyping, to a situation where we harvest what's in the electronic health record and utilize opportunities to do a very rapid neurobehavioral assessment that can be conducted virtually for at least some of these patients, and to put that information in a data commons. And like, this all sounds kind of magical, but the reason that we can do this is that within the context of the COVID pandemic, there were mechanisms for developing data commons like this related to COVID to rapidly assimilate information about the patients who were streaming into the hospitals, and using data platforms that could retrieve information from Epic and Cerner-based electronic health record systems. And we basically just pirated that technology and asked, could we do the same thing, not when a patient has COVID, but when they've got a variant of unknown significance in a brain gene? And so that project is off the ground, it's running. The IDDRCs are helping by doing some of the phenotyping of these prospective patients, but they're coming from all over the medical environment that of patients who've been clinically genotyped. And this would be a way of accumulating lots of data to help those individuals who have variants of unknown significance have their data, not only their genotype, but their phenotype coupled with other patients, and then migrating critical masses of that information to the gene curation process of the NIH and seeing if we can get those families more information about whether those variants are actually causal for those conditions. Finally, COVID-19, and one of the great health disparities of all that's been discovered through the accumulation of health records information. This is data from over 64 million patients across 547 healthcare organizations, the bottom line of which is that if you ask of all the established patients in these health systems, what is the principal risk for COVID-19 mortality, intellectual disability ranks way up there as one of the predictors of mortality for all patients. And so COVID-19 has impacted this population, not only when they're infected, but when they're uninfected, because of the impact of the COVID-19 pandemic on individuals with autism and related disorders, in the sense that the mitigation procedures, the isolation, the loss of professional therapies and in-person supports has really devastated this population. And methods to not only manage the hardship and the stress of this social isolation on this population, but also to get children affected by these conditions, even when they need very up close and personal intervention, and get them back in school, has been a major important effort to respond to this particular very cruel health disparity. One of the ways that we're doing that among the IDDRCs is to implement rapid saliva testing for students with developmental disabilities and their teachers, to get everybody very rapidly and frequently tested to ensure safety among the students. And that's resulted in comfort of these educational systems with getting these kids back in school and working with them in even very close quarters, and also making sure that that the treatment community recognizes how to manage the behavioral consequences of the impact of social isolation and the loss of therapy services and some of the chaotic circumstances of busy households that are now the playing fields for day-to-day life for the kids and their education. That the behavioral consequences of these kinds of stressors can be managed by the clinical community, where it's often not that many people in the community that know how to manage dual diagnosis, that is developmental disability coupled with behavioral decompensation or behavioral disability. This is an article that accumulates from the scientific literature practice parameters for addressing those conditions. And recently that has been actually distilled into a smartphone mobile app for clinical decision support for clinicians that you can actually pick up just by typing into the App Store Missouri Alliance for Dual Diagnosis, and check out that app. And this is the sort of scholarly article on which a lot of the information in that app from which was distilled. So I'll stop there. This is a graphic of our scientific team at Washington U, especially the group that really has involved itself with the exploration of these disparities related to autism. And I'll look forward to an opportunity to see you and meet with you live in the training day to have a discussion about any of these particular disparities and the topics that were addressed in this presentation. Thank you very much.